Hello, dear students. We will start the biology assignment and welcome to the today's class. 20th week assignment for English version. And it was a very most easiest assignment we have got in our any previous kind of assignment from biology. And you will see here the selected work for the assignment. Just for a couple of lines is mentioning there that is during the cell division the chromosome can be changed in different stage and mark it draw this figure and just mention the stage with changing stage of the chromosome in different stuff mitosis cell division. Very simple. You can use the poster paper. If it is not possible for drawing that one, you will use the opposite side of the calendar and you can get help from the textbook. So during cell division, we know that mitosis cell division where the chromosome can be changed frequently according to the standard of class 9. So let's go directly to the solution of that class specifically. And very first time, and how can you get the good marks that mentioned here? Each and every stage you can mention where the change is occurred, and then you can write some features with figure, and then you are actually present in your excellent way. So let's go to the solution. But first time we give the definition of cell division. We know about cell division, starting part on there. Division of cell. Normally we can write in there division of cell. In which way? In which way one cell divides frequently to produce numerous number of cells. Numerous number of cells by a process is called cell. That means we will go through the definition very first time since it is asking for the topics of cell division. So division of cell, in which way one cell divides frequently to produce a number of cells by a process is called cell division. Normally whatever we have written in there, and our assignments say that how can we change that chromosome in different steps of cell division. We know that mitosis cell division, in mitosis cell division we can write accordingly. Mitosis cell division, sorry, in mitosis cell division the change of chromosome, changes of chromosome observed. Mitosis cell division, change of chromosome observed in its different stage. So we know about the mitosis cell division is categorized in two stage. First one in the karyokinesis, and second one is the cytokinesis. We know about that kinesis. We mentioned that one, but very first time we will explain here all the phases, likely the prophase, then prometaphase, metaphase, anaphase, and telophase. telophase. Okay, so listen, since the changing of the chromosome are mentioned to be here, so specifically we will mention the karyokinesis of the mitosis cell division, because we know that mitosis cell division are of two kinds and two types we have discussed in before during the class time. And it is a karyokinesis and cytokinesis. Cytokinesis is actually a division of cytoplasm where chromosomal change is not in there. But in case of the karyokinesis of mitosis cell division, that is to be counted. It is important, very important for to know that all the five phases we will discuss. And before that, when I start the prophase, before we will go through about the interphase of a preparational phase of nucleus, then other all these five phases actually after writing this one we will mention in the poster paper as a project, or in the opposite side of the calendar. That would be better for having the change of chromosome in different way and different stage. Okay, so let's go to move on there the real explanation in case of the assignment what is asking for, and we will try to go through on there. So let's move on the prophase, but before prophase you will better for getting good marks if you mention the interphase, another though it is not given in your book, but a preparational phase is also mentioned here, likely to have. I write there the interphase. Interphase, you know about that, the interphase. It is the preparational phase. It is the preparational phase. Preparational phase of nucleus to start cell division. To start cell division. Cell division. Chromatin fiber. Chromatin fiber. Spread it. Spread it all over the nucleoplasm. Nucleoplasm. And we will just follow me to draw that one. Just this way. I just try to make it figure in this way. This is, you can better of following the drawing of that one. The nucleus and the chromatin fiber. Normally inside the nucleus we have seen in there. You will mention accordingly. This one is the cell wall, then plasma membrane, then nucleus, that means nuclear membrane. I have written in one case, you will write in each and every cases when we will go through the prophase after then prometaphase, likely that way. Nucleoplasm, then chromatin fiber, then nucleolus. I have written for one case, but you will write this figure interface. Better to mention the very first time the interface, because you know that the interface is that phase start the cell division after going through the nucleus formation. So it is called the preparational phase of the nucleus to start the cell division, where the chromatin fiber, these are the, we have seen that we told that it is a chromosome, but before starting prophase in the interphase, it is look like a thread, like a structure twisted here and there, and likely to have seen, and we can tell this one is a chromatin fiber. Okay, this one is known as chromatin fiber. Okay, so it is the chromatin fiber spread all over the nucleoplasm. We have seen inside the nucleus the liquids are in the nucleoplasm, and these are the cytoplasm inside it. You can see, you can write in the cytoplasm. And I have mentioned here specifically the cell wall, plasma membrane, nucleoplasm, nucleolus, chromatin fiber, cytoplasm, and interphase. And each and every case, when we go through the drawing figure, we will mention that one. So very first one, this one we will explain up, and then we will go through the prophase. Sorry, dear students, electricity gone, so maybe the shortage of light we have seen in there. But no matter, since I have seen the board in the class, and I think you also have seen this one, though it is the less light in the board, I think it would be manageable. Okay, let's go to move on there. I will draw for one figure here specifically. Uh, we will cover this one with the nucleus. You can write accordingly or draw likely that way. Okay, the nucleus will be on there. And look at here, it is very important to know that how many uh, chromosome I will start with then. I will start with the 4 chromosome. 4 chromosome in there. Centromere with chromosome. That was not finding in case of the interphase. Interphase here we get the chromosome, because in the preparational phase where the thread like structure of the chromosome, and we say that is the chromatin fiber, and here we actually got the 4 chromosome in there. Okay, 4 chromosome I have drawn in different way, or any way you can draw likely that, and you must mark that one each and every part, since I have mentioned each and every part in before, or likely that way I didn't write in there. Figure prophase, figure prophase, or likely that way, prophase. And not only that one, we will go through the features. Prophase we will write in there, prophase and features. Prophase and features are likely to have in there. Listen to got that point. Whatever we have seen, we will write on there. The well-developed nucleus are found. Well-developed nucleus. Nucleus are found in there. Second figure, a second point of features we can write accordingly. The chromatin fiber, chromosome frequently lost water, and become shrunken, and at the end of the stage, you can write in there, at the end of the stage, nuclear membrane start disappearing. So the assignments say that change of the chromosome, we already counted here the change of the chromosome in different way, likely to have interphase. It was a chromatin fiber. Now we got the getting a shape of the chromosome in there. And in the prophase, first stage, feature well-developed nucleus, whatever we have seen in the board. In case of the division stage, I will write accordingly, since it is a well-developed nucleus and chromosome in the interphase. After then, when I start with the prophase, they frequently lose their water and become shrunken, just concise on there. When the loss of the water, they become shrunken on there on the time. And at the end of the stage, very end of the stage, nuclear membrane, the outer layer of the nucleus, this one just start disappearing, not yet disappear. In this next stage you have seen it will be disappeared, or likely that near about disappeared on there. So this is, you will draw a figure, mark that part, each and every part, and then write three features. That is better to mentioning the different stage and different changes in the chromosome in the first stage. So this is the prophase. We will go through the pro-metaphase. Next one, pro-metaphase. Pro-metaphase. We will write accordingly the pro-metaphase, and you will see here the figure we will draw. You must draw each and every single figure different way, but I have drawn this one so easily I can erase and draw the inner part. So we will turn out the two phases in there, and we will just try to draw the figure this way. This way, and look at here, the chromosome will be attached in the chromatin fiber in different way, likely this way. This way, just for having the different shape we can get in there. And the chromatin nuclear membrane, you can see in there. It's better to draw concisely with the pencil so that it will be better. Nuclear membrane and nucleolus, you can write in better. The nuclear membrane and nucleolus in previous one. Nucleolus, nuclear membrane and nucleolus start disappearing in the last stage of the pro-metaphase. So second stage, whatever we have seen in the figure, we will write accordingly the features. Okay, pro-metaphase. So what is the pro-metaphase say? We didn't see in the prophase this structure, but now we have seen a different structure. These are the, you must mention that one. This one is the pole is mentioning in there. And equator region, you can write in there equator. This is the equator, pole and equator. So spindle apparatus, this is known as the spindle apparatus. This structure we have seen in there. So spindle apparatus with two pole, spindle apparatus with two pole, spindle apparatus with two pole created here, created this stage, in this stage, and centromere of the chromosome, look at here. Two pole means this two pole is apparatus like thing we have seen in here. So it is called the spindle apparatus created here. Centromere of the chromosome, centromere of chromosome, centromere of chromosome attached with the fiber of spindle apparatus, spindle apparatus known as spindle fiber or chromosomal fiber, you can write in there, known as spindle fiber or chromosomal fiber, we can write in there. And then after, nuclear membrane and nucleolus near about disappeared. In this stage. So three features, finding some important features from each and every stage of the biology cell division. So it is to be better to have written in there. We have seen whatever you have seen in the figure, that is to be the features in here likely to have. So spindle apparatus with two pole created in this stage. Centromere, you have seen the centromere of the chromosome attached with the fiber of a spindle apparatus is known as a spindle fiber. You can see the traction fiber or chromosomal fiber, and nuclear membrane and nucleolus. Nuclear membrane, look at here, the nuclear membrane, it was very clear in the previous stage, but this stage are just not be properly found in there, so it is near about disappeared. Spindle, inside the spindle apparatus, nuclear membrane and nucleolus near about disappeared. Three features. And we have seen the chromosome on there in the previous stage, the changing of this one, and now we will see another way in the metaphase what are the changes of the chromosome specifically for having in case of the cell division, in case of the mitosis cell division here. Okay, let's go to the metaphase stage. I think you understood this one since I taught you in before during class time, and also having likely to have assignment. Since the assignment, it is you must use the poster paper or opposite side of the calendar for make it bold. You can use the sign pen for drawing that one, and better for having a good visuality, drawing a single figure, marking their part, and features that would be the better cutting the good figure in the assignment. Okay, the third one, metaphase. So it is easy for me to just erase the inner part. You must draw the figure, each and every figure in your copies or like that. So one pole to another pole, I will ensure of this one. But this time the apparatus are different look. And we have seen this time the chromosome is likely the different than the any other stage. And their position will look like different in that way. Okay, and I'm sorry, I'm once again I'm sorry that the electricity corner less light I face in there. By the way, we will complete the assignment here. Oh, sorry. This one is the prophase I have written in pro metaphase. Each and every figure you will write below the figure. Now this figure is the metaphase, and before you will write there the pro metaphase or likely that. So what we have seen in there, in case of the pro-metaphase, and now it is the metaphase. Number third is the metaphase. Metaphase. So we have seen the form spindle apparatus. We have seen specifically, and the features. First one, chromosome. Chromosome in this stage. Become maximum short and thick, and you can see in this stage chromosome are getting become maximum short and thick, and centromere of the chromosome, all the centromere placed parallel in equator region, and nuclear membrane. You can write here nuclear membrane and nucleolus completely disappeared. No nuclear membrane, nucleolus are found in there. Three features we have found in the chromosome in this stage become maximum short and thick. Metaphase, maximum short and thick. All the centromere placed parallel in the equator region of that spindle apparatus. You can write better on there. and there is no nuclear membrane and nucleolus. That means nucleolus and nuclear membrane completely disappeared from here. So this is the metaphase. You will write in the metaphase, and then we will go through the anaphase. In the case of next one, that is the anaphase. I will draw the figure accordingly, just for I erase, but inside the one figure I try to draw all the figure. You better each and every single figure you will draw in there. And look at here, this one is also being there. 1 2 1 2 1 2 1 2, likely that. The change of chromosome, we better understanding that what type of change is actually found in this stage, or all of the stage we have mentioned here. This is not all the chromosome are greaterly found in each and every cases, every figure. So this figure is the anaphase. Anaphase. So it would be the anaphase. Anaphase, and it would be the 4. Anaphase. We'll see some features in different way, likely to have what is the features actually mentioned in there. Anaphase. So the centromere divided, whatever we have seen in the metaphase, all the centromere divided. Okay, centromere of chromosome, centromere of chromosome divides and start moving toward two opposite pole in this stage chromosome look like English alphabet I, J, L, V, etc. when centromere reach at two opposite pole the stage completed. Okay, I have written 3 features, then I will tell you the simple, very simple features we have written. Chromosome divided, it was 2 strand in 2 opposite phases. We have seen in case of the metaphase, but when it's divide, one part likely that one, this one to be there, but half part, now half part is upper hand, half part is lower hand, likely in the spindle apparatus. And look at here, all the centromere are trying to move on to go there to opposite pole. Two opposite pole means this one is the pole, this one is the pole. So this centromere move, and chromatid followed by it move. They are divided and try to move, try to reach at two opposite pole, start moving. And in this stage, the chromosome look like IJLV. This look like I, this look like J, this look like L, this look like V. Though I have drawn the all of the figure V like, but actually in this stage, the chromosome look like IJ, LV, this shape, English alphabet in this stage. Okay, and then after when centromere reach, they start moving. When they are reach of their two opposite pole, the stages end up and completed. Okay, so this one is the anaphase stage. And finally, we will go through another last stage, that is the telophase. Telophase, let's move on there. Telophase. Okay, if the line is being cut, figure very quickly here, since we have very few minutes in our hand. Features, whatever we have written in there. Look at here, the chromosome is now in the previous shape, whatever we have seen in the prophase. So two daughter nucleus, not cell actually, nucleus, daughter nucleus created in this stage, and then after we will write in there nuclear membrane, and nucleolus evolve once again, and chromosomes frequently gain water to become expanded again, or likely to have, we have seen. So two daughter nucleus created here. We have seen in there nuclear membrane, nucleolus once again evolved, and chromosomes, whatever we have seen in previous stage and very first stage, lost their water, but here gained their water and become expanded once again. These 5 stage you will draw in the poster paper